today we're going to have, for this hour, a lively discussion, we hope, of what's at stake for the world as a result of the new activities in biological sciences, particularly in genetics. Those of you who heard the aperitif that David offered on Thursday know something about how what he's going to talk about is situated within the history of genetics. And we're going to talk today about some of the practicalities, some of the implications for science and for medicine. But before we do that, let me give you a brief synopsis of what I, as your grateful moderator, plans to do. I'm going to say a couple of words about our speaker, my friend and colleague David Baltimore, and then we're going to have a talk by David laying out some of the principles of what he referred to in the aperitif as CRISPR-Cas9 and its potential for changing the way we do biology and think about our genetics. Then David and I will talk about some of the things that he mentioned during his talk, and we will leave a hearty amount, hopefully 15 minutes or more, for questions from you. Hopefully not questions that address your private medical situation, but questions that raise grander issues. Neither of us practice medicine. I don't practice anymore. David never did. And we will defer to private discussions what the consequences of CRISPR might be for your own problems. David Baltimore is a name probably known to most of you. He's one of the giants of 20th century and 21st century science, known for work in three great areas. One is doing his own research and training a number of people who are now esteemed scientists throughout the world, perhaps best known for his discovery of the enzyme called reverse transcriptase, which explained how retroviruses, which include AIDS, work, and how and became a staple of the biotechnology industry and of everybody's day-to-day work in a laboratory. Analogies to that may be appropriate when we talk about CRISPR, and for the discovery of reverse transcriptase, he was awarded a Nobel Prize in 1975, a mere 41 years ago. He still looks good. But while the reverse transcriptase discovery was notable, David has also made tremendous contributions in the study of gene expression, how the immune system works, to virology, and to many other aspects of modern biomedical science. He's also been an important leader of many institutions, the founding director of the Whitehead Institute at MIT, the president of Rockefeller University and then Caltech, and where he is now, as you heard if you were in the previous session, like everybody else here, a member of the faculty there. And the third, and most important perhaps for today's discussion, he has been a leader in discussions of science policy on a national scale. Those discussions began as early as 1969 when he was a new assistant professor and led a study of chemical and biological warfare with a group of graduate students, but then moved on to being a leader in the discussion of the implications of recombinant DNA technology at Asilomar in 1975. He was an important spokesperson for HIV/AIDS research in the mid-'80s, and very recently was the leader of an international meeting that was held at the National Academies of Science to talk about some of the implications of CRISPR technology. With that, I think we're ready to, the flickering has died down up here, and I'll let David make a presentation that will illuminate for you many of the things he tried to use to entice you to come here in the first place.
Thank you, Harold, for your kind words. Thank you all for being here. Thank Donna and Ben for making all this happen. I start with this comparison between science below in Darwin and Morgan's days and science above today. Make of it what you wish. So if we go back, as I said in the aperitif, if we go back to the 19th century, we had absolutely no idea that the world changes, that our genes change, that life is a continuum. And that all changed with Darwin and Wallace, and then with Mendel. And we learned that genes were underlying heredity, that genes could change. And what's happened in the last century, really, is that we've gone from understanding that there was an underlying substrate of biology to understanding what that substrate is and to being able to consciously alter it. And that's an enormous change, especially over a relatively short period of time. It really dates from 1953 when Watson and Crick discovered the structure of DNA, because when we knew the structure of DNA, we could start thinking about how DNA works and ultimately how we can alter DNA. So the double helix is the real starting point for what I want to talk about. And after that, we still couldn't read DNA, we couldn't alter DNA. We had to learn how to do all those things, and it's all chemistry. So it was a change of biology from being an observational science to being an arm of chemistry. And we've been getting these capabilities year by year, each producing its own little revolution. And overall, we've reached the point today where we now have the ability to alter genomes with the ultimate precision. And I'll show you what that means. And it came about because in 2013, a number of scientists figured out how to take what was a natural capability of bacteria and adapt it to use in mammalian cells, in human cells. And I'm not going to go into the background of it. It's a wonderful story. But fundamentally, Emmanuel Charpentier was the inheritor of a tradition of working with CRISPR-Cas9 in bacteria. She came over to the United States and worked in Jennifer Doudna's lab at Berkeley, where they figured out how to make biochemistry of this. And then Fen Zhang and George Church figured out how to adapt that so you could actually put it in a cell and get it to modify DNA in a cell. So how is that done? What does it enable us to do? What do we expect to be able to do soon? And what should we do in an ethical sense? Because I think that's the largest question that will face humanity as time passes. So how does CRISPR-Cas9 work? What really is it? The key to altering DNA in a cell is to break that double-stranded DNA right across, a double-strand break. And the key to it is a key because it's a very dangerous thing for a cell to have happen to it. And cells abhor broken chromosomes. And the cells mobilize this repair army to fix the break. And in the process of fixing the break, there is the opportunity to modify the DNA sequence. So this is now a real piece of DNA with all the chemistry, and I'm not going to go through all the chemistry, but I will point out that it is this pair of bases that span the double helix, and here they are opened up, which are formed of nucleotides. And there are only two such units in the whole DNA. One puts C with G, one puts T with A, and of course you could have the inverse, A with T and C with G, or whatever the inverse is of what I said. And that means that this is a very nice, smooth structure because these two base pairs, nucleotide pairs, have the same fundamental size, even though they have a different structure and therefore a specificity. So to make a DNA alteration in a cell, what we need is two things. We need two things first and then a third. The two things we need is something to cut right across the DNA. We call that a nuclease. And something to recognize the position along the DNA. So we have 3 billion base pairs of DNA, and we have to be able to scan that whole 3 billion base pairs of DNA and find one spot if we want to alter a genome in a very defined way. So CRISPR is an agent that allows you to find a spot, and Cas9 is a nuclease that cuts. And then we need a third thing, a donor DNA, and I'll show you why that is. So here's a chromosome down here, and CRISPR has found that chromosome, that piece of chromosome. CRISPR has a sequence of nucleotides. The DNA has a sequence of nucleotides, and those sequences are exactly complementary to one another. So you get a nice little double strand of actually, it's an RNA with a DNA, but it doesn't really matter. And that then takes Cas9, which is the eminence grise in the background here, and tells Cas9, cut the DNA in an absolutely defined position. And the rest of this is just to hold it all together. So here it is again, Cas9 cutting DNA. Here's the broken site in the DNA. And now the cell mobilizes this army of enzymes to fix that break. If it simply fixes the break, it makes a mess of it, because it adds a few nucleotides, it takes away a few nucleotides, and you get an altered gene which won't function. So that's not a great outcome unless what you're trying to do is to knock out a gene, and we do that experimentally all the time. But if you provide a donor piece of DNA that has exactly the sequence across this break, that donor piece will act as a template for correction of the DNA and for putting back together again the broken ends. And in the process of that, you can fake it out by putting a single base pair change in the donor, which will then be carried into the chromosome. Or in fact, you can put in a lot more than that. You can put a whole gene in. And that's the alteration which allows this system to modify any one or any combination of the 3 billion base pairs in your DNA. So let's take a look at a case in which there is a typographical error in your DNA. It causes sickle cell hemoglobin, sickle cell disease, and it is a single change from an A to a T in the gene for part of hemoglobin. And now we want to correct that, and that's exactly what CRISPR-Cas9 can do. Correct that and do nothing else if we have it under complete control. The example below is not important. So what can CRISPR-Cas9 do? It can inactivate any one of the 20,000 genes in a genome of an experimental animal. We do that all the time. It's extremely useful. We can correct any mutation, altering as few as one base pair in the 3 billion in a cell. It can add genes to cells. All of that we call gene editing. It can actually also change the function of genes, and CRISPR-Cas9 is just one example of a whole family of proteins that are being examined now. So this is an ongoing research effort and will continue for many years. Now, in humans, all of us have sections of our genome which are not optimal. Some of us, unfortunately, have ones that are highly deleterious. And it has been a dream of medicine since the notion of genes came about, that you could change those genes back to the normal configuration, the average configuration that most people have. That's what it would mean to fix a problem, to edit it back to normal. Now, in our body, we have trillions of cells. If you wanted to do that in every cell, it's not possible. But there are ways of using CRISPR-Cas9 in tissues which have stem cells. So this is a stem cell. this is the blood stem cell, and that stem cell can give rise to all of the elements of the blood: red blood cells, platelets to stop clotting, white blood cells, which are basically your immune system. All of those derive continually through our whole lives from blood stem cells that are held in the bone marrow, which is where blood comes from. And they are the cause of diseases. Sickle cell disease is because of red blood cells. The bubble baby disease is because of immune cells not functioning. There are platelet disorders. All of those could be corrected if we could use CRISPR-Cas9 in the blood stem cell and get rid of the disease stem cells and replace them with normal. And that can be done and will be done and is under development now today. That we call somatic gene editing, because we don't pass that on to our children. But we have in our bodies single cells, germ cells, single types of cells, germ cells, that we do pass on. And of course, to form a child, you bring together a sperm and an egg. And that allows us, at that moment, to alter the genome of our children. And there is where CRISPR-Cas9 becomes a very powerful technology and a slightly scary one. So how do you do that? Well, you really have to do it in a petri dish, in a test tube, in a laboratory. You have to take eggs from a mother, sperm from a father, put them together in a laboratory, get an in vitro fertilized egg, and that then is the target for CRISPR-Cas9 in order to modify an inherited gene. So if there was a sickle cell gene in this combination, you could correct that back, at least we could imagine, we can't do it today yet, correcting that back to normal. Now, we can then allow this to grow up to the 8-cell stage, and at the 8-cell stage, you can then take out single cells from these embryos and check to make sure that everything was done right. And that means that you can put into a uterus to turn this into a child a fertilized egg, which we know has been altered in one specific place and nowhere else. And so we find the embryo that has the right genes and we put that in. But do we really need to do this? Because if we're doing in vitro fertilization, then we can use something called prenatal gene diagnosis, or PGD. And that will tell us which one of the embryos has received bad genes to start off with. In most situations of genetic disease, and a husband and wife who come together, there are good genes as well as bad genes. And if you can find the embryos that have the good genes, just using PGD, you don't need to alter the genome. And so we can find that this and this and this have perfectly normal genetics and use those to generate the next generation. Because of that, the actual need for genome editing is small. It's not zero, but it's small. Now, everything I've talked about up to now is rare Mendelian traits, traits which are due to a single gene. Actually, most of our genetics is polygenic, involves many genes, and the common diseases like heart disease, schizophrenia, obesity, and traits like height or intelligence are all determined by multiple genes. And those multiple genes work together, they work apart. They're going to be very hard to modify. So decreasing disease risk or enhancing the human population by, for instance, trying to affect intelligence will be difficult, but not impossible. And again, over time, I can easily imagine that it will be happening. So because we can imagine this over time, we called a meeting in Washington, which Harold referred to, sponsored by the U.S. National Academy of Sciences, the Royal Society of Great Britain, or what's left of it, and the Chinese Academy of Sciences. And I was the chair of a 12-person international organizing committee. We met for 3 days. We had ethicists, geneticists, medical doctors talking about this. And in the end, we said it would be irresponsible to proceed with any clinical use of germline editing until there's a broad societal consensus about the appropriateness of the proposed application. I assumed that that consensus would be there for something like sickle cell disease, might not be there for more complicated issues. And so we called on the academies to continue this discussion, and they will continue the discussion. My opinion is in two forms: what will happen and what should happen. What should happen is we should perfect the methods and be thinking about this carefully and not rush into anything. What will happen is that there will be a physician somewhere in the world faced with a very difficult case in which editing technology is the only way that a couple can have a normal child. And for that patient's sake, this physician will find a way to start a revolution. It's going to produce headlines, it'll produce hand-wringing, but it'll be an opening wedge, and it may well not occur in the United States. So, to wrap this up to now, the genetics of our children has been determined by a lottery, initiated by sex. Whatever genes you get, you get. In principle, we now have the ability to control our genetic destiny. Do we want to leave our destiny to chance, or do we want to take control? Are we smart enough to do this well? And remember, each decision that we make affects not only our children, but a whole lineage of children that will be born, stretching forever. Thank you.
David, have a seat.
Yes.
So in my conversations about CRISPR, the three things that tend to raise, can we raise the lights without flickering?
I'm afraid to ask for lights.
Yeah, we don't need the darkness at this point. The three things that always come up and create the most exercise conversation are, first of all, should we use CRISPR and the germline, something you've discussed and I'm sure members of the audience will want to ask about. Secondly, can CRISPR be used as a tool of bioterrorism, something we might want to say a few words about. And the third question is always, how much money is there in this and how do I make it? But nevertheless, for the average guy working in a laboratory, my postdocs, your postdocs, the big thing, though there are a couple of big issues here. One is, how can I most effectively use this technology to answer the kinds of questions I want to answer? And this brings us back to other kinds of discoveries, your own discovery of reverse transcriptase, which did explain a big biological problem, but also provided the tools with which the biotech industry got going. People like me began to work on cancer genes. Very, very important problem affecting the texture of science. The other big issue, one that I'd like all of you to take away from today's exercise, is that CRISPR is a major discovery that came not just because the four heroes of CRISPR that were shown on David's slide ran their big laboratories, but in part because there were folks 15 or 20 years ago who were trying to understand some bizarre nucleotide sequences they saw in organisms. Some of these people were working in yogurt factories and studying lactobacillus that is helped to grow yogurt. And they saw that there were sequences in the genomes of these organisms that looked like the same sequences found in viruses that invaded those organisms. And thinking through this mystery led to a great discovery. And this is the kind of research which in days of impoverished funding of our major agencies and the difficulty of getting funds for doing the kind of I hate to use the term because it's so often criticized, curiosity-driven research leads to the great discoveries, but is less likely to happen at a time when we all say, let's take the next step forward to curing whatever disease my family has. So these are major dilemmas, and I think for all of us who do biological science today, CRISPR has transformed the way we work, and that only happened because of the very esoteric, even Baroque story that led to these discoveries. So maybe you could say a couple of things about the backstory here, or would you rather say a word or two about the unmentioned topic of bioterrorism, which I firmly believe is not a major likely consequence of the use of CRISPR, but people do seem to ask about it wherever I go.
Yeah, I know. And I don't understand why either, to tell you the truth. The revolution that enabled us to manipulate DNA, we call the recombinant DNA revolution. And that really was, the signal event was in 1974. After that, there was a great concern about bioterrorism. The words weren't there yet, but biological warfare. Because suddenly we could manipulate genes, and we could manipulate the genes of bacteria and make them more pathogenic. That was at least a thought. The truth of the matter is that certain organisms are lethal to us, and they're highly evolved to be lethal to us. And you can't do a whole lot better than that. And it really has been largely unnecessary to do any better than natural organisms. With CRISPR, we've come to a new level of sophistication in being able to modify genomes in situ, in bacteria or elsewhere in viruses. But I don't think it changes the picture a whole lot from where we were in 1975.
Yeah, that's my own sense that nothing more virulent in the world than a smallpox virus or an anthrax spore, and you don't even have to deploy these agents. You have to say, I have a vial here with this agent,
right.
And that creates the panic that bioterrorism aims to achieve. And the notion that CRISPR is going to improve on that is really fallacious in my view at this point.
But it is a worry of the government.
Yeah. I'd like to get back for a minute to this question of our ability to change organisms or cells, because CRISPR is not the first vehicle for doing that. We've had the ability to introduce genes into cells, not to edit the cells, but to introduce genes for some time. And I think this was appreciated as early as the time of the recombinant DNA discussions. Some of you may remember, if you were readers of the Rolling Stone in 1975, that Hunter Thompson, the gonzo journalist, had a headline that emblazoned his story about the Asilomar meeting, saying, now that we know how to manipulate the genetic code, what are we going to say? And I think that's still an important question. For the average experimental scientist, the issue is, from someone like myself, how does a normal cell become a cancer cell? And then there are a series of things we'd like to do to cells to try to explore various questions about how cells become cancer cells. And for that, we either insert something important into a cell, or we try to use CRISPR to change the behavior of cells. David went very quickly over something that's truly amazing to many of us, that it's now possible to make what we call a library of viruses that can infect a mammalian cell, and in one infection, directly change the nature of every single gene in a collection of cells, and ask the question, what gene is responsible for some biological property of the cell that we're observing? This is a power of what we call high throughput technology. It was unimaginable just a few years ago until CRISPR came along. And the way in which all of us work has been fundamentally changed by this set of discoveries.
But I emphasized the potential role of CRISPR in human gene modification, because I think that's the only aspect of it that raises questions that a larger audience is going to be concerned with. You don't care how Harold's postdocs solve their problems. You just want to know the answers.
Oh, there are a few that care.
There are a few.
My wife wants to move it along more quickly.
And so that's why I focused on that. But the utility of CRISPR in the laboratory is just amazing.
I think when we talk about the treatment of disease, I think hope you understood from what David had to say that the very strong consensus among responsible scientists that we're not ready to take this incredibly monumental step of going into the germline at this point. But we are ready to think about making changes in genomes using what we call somatic cells, cells that don't contribute to the germline. And for those diseases like sickle cell, perhaps thalassemia, and possibly some others, possibly ways to alter the immune system to make individuals resistant to HIV, something you have been worrying about in your own scientific life. There is the potential for changing some of the cells in our body that don't contribute to the germline in ways that could be influential in the way that gene therapy has started to be influential on a relatively small scale, even before CRISPR came along.
Right. Gene therapy is a way of providing genes which for some reason are not functioning, some inherited reason generally. And the bubble baby is the classic example of it. Children who are born basically with a non-functioning immune system because of a single gene which has a single alteration in it. And we've been able to...
Maybe we should explain the bubble. So these children live in a plastic tent, resembles a bubble, and are protected from external
Right, they can't be touched by anybody. They can't have any normal social interactions. It's a terrible way to come into life. But so the answer is to give them the good gene that they're lacking. And we've worked on that problem. Actually, I have worked on that problem, but the whole scientific community has for many years, and have found that we can make viruses the carriers of genes into cells, and we can correct the defects for many of these immune deficiencies. But what we're doing is we're sort of randomly putting genes back in. What we can do with CRISPR now is surgically alter a bad gene into a good gene. That is so much healthier from the point of view of the organism that it's going to be a huge change. It'll take some time before it gets into use. Meanwhile, we're using the somewhat clumsier but effective techniques, and they are now in clinical use in 1 or 2 cases, and will be more and more in the near future.
And one way to illustrate the potential for using somatic cell gene therapy or CRISPR treatment to treat diseases comes in my own field of oncology. People frequently ask me, well, with CRISPR in hand, can you now cure cancers? And the fact is that it's really impossible to fix the genetic damage that gives rise to cancers in every cancer cell with CRISPR technology as we know it today. But it is possible to think about using gene modulation of cell behavior, either by introducing a gene randomly or by editing more precisely to, for example, booster the immunotherapy of cancer you've been hearing about, or to make bone marrow transplantation more likely to succeed by adapting the bone marrow that's going to be transplanted into a cancer patient into a more acceptable transplant. I see that the clock has given us 15 minutes left. At this point, I think I'd like to draw to a close the private conversation, invite any of you who have questions about any of the things that David and I have been talking about to raise your hands and speak. I see a hand right down here near the front. Wait for the microphone, please. If you just say your name and try to make your question a question, not a comment.
I'd love to hear more about the possibility in treating HIV because of wouldn't it be possible to deal with more T-cell production or something from individuals that are resistant to HIV infections? You know so much of.
Yeah. You remember the slide I showed of the blood stem cell giving rise to its progeny, something that continually happens. One of those progenies is T cells, which are the cells in which HIV grows. And there's a receptor on the surface of the T cell which is necessary to allow HIV to get into that cell. If there's a mutation in that receptor, then first of all, it doesn't really seem to be necessary for any important normal function. And second of all, the cell becomes resistant to infection with HIV. Fabulous. So what we want to do is to take the bone marrow stem cells and knock out the receptor gene for HIV and then put them back in a patient and make his or her immune system resistant to HIV growth. That's all doable. It was actually doable a number of years ago, and there's a company that's been trying to develop it clinically, Sangamo. And with CRISPR, it's just a lot easier now to think about doing it. We've been doing it by using an siRNA, a negative knockout of the gene. That is now in extensive clinical trials and is looking interesting. So there are a number of ways that we're going at just that one receptor to try to make cells resistant to HIV in otherwise infected people.
So the goal there, of course, is to allow the cells that can't be infected to prosper, even if other cells end up being killed by HIV. Questions down here in the front. I see a couple hands. Right, third line.
Hi, my name is Rob Rosen. I'd like to ask a question about sort of the concentration of ownership of great research in the hands of big pharma and the sort of relationship between great teaching hospitals and universities and big pharma, and whether that's good for the development of the breakthroughs of the next decade that will transform all of our lives, or whether that relationship is it a positive or is it a hindrance to the evolution of great science?
Are you speaking generally or specifically with respect to CRISPR technology?
I was trying to broaden it from CRISPR and perhaps also get it into your area, Dr. Varmus, in oncology and in immunology, but I was trying to broaden the question beyond just CRISPR to a broader question of just the state of great research and its relationship to big pharma.
David?
It is generally felt, it's generally said, that the patent system, which gives a monopoly position to somebody who is the first discoverer, and I might say with CRISPR that's now a big matter of debate with the patent office, that is the most effective way to move things out of the laboratory into actual clinical practice. And I believe that. And I know that, because I'm a director of Amgen and have been for many years, which is one of the big biotechnology companies in the United States, that the amount of money it takes to get something from an idea to an actual drug is billions of dollars, literally billions of dollars. The amount of money that it takes to actually discover things is millions of dollars. So there's a huge disparity. And for a company to put up that amount of money, they've got to have an idea that they have a market available to them. Otherwise, it just isn't, there's no way of getting investment money. So I think that system works. And the Bayh-Dole Act, which gave the rights to universities of their discoveries and allows them to license companies to exploit the discoveries, has worked pretty well. there are times when a new tool comes available and you don't want to see it all tied up with one pharmaceutical company or one licensee. And that's the sort of downside of the patent system. And I think with a tool like CRISPR, you wouldn't want to see that be available only to one company. But in fact, it has been licensed. It's been licensed by a number of the discoverers whose pictures I showed, to 2 companies that are startup companies. And I think that may be a problem. I don't know what's going to happen as we go downstream with that. But I am certainly an apologist for the patent system.
Yeah, well, I agree largely with what David had to say, but let me just add a couple of things. One is that I do think that CRISPR technology will be broadly licensed, in part because there are going to be competing ways to use CRISPR-like technology. CRISPR is being improved. It's not the end stage. There are other CRISPRs than CRISPR-Cas9 that look like they may be improvements. One of the things that David mentioned, but I think needs emphasis, is that right now, we can't be assured that when we make a gene edit with CRISPR, that there aren't other changes occurring elsewhere in the genome. That's one of the reasons why many of us feel very conservative about going into the germline. And making sure that other things are not happening is a very important feature of all this, and some of the new systems are going to have higher safety requirements. The other point I think is self-evident for those who were looking at drug pricing is that although patents are limited, even in that limited time, very, very high prices for certain drugs have become a serious problem that I don't have a simple solution to. But if you look at what's happening in this dynamic marketplace at the moment, there are places, particularly in oncology, where some of these drugs are successful only a small percentage of the time, that the companies are going to be considering the possibility of charging only in those situations in which the drugs are used with good outcome. So that would be one way of thinking about how prices would be modulated. Question I see halfway back, and the lady in the front has been patient. So, lady with the green scarf. Thanks.
Thank you. Do you see CRISPR being affected in the entire arena of autoimmune disease?
I think autoimmune disease is one of those areas where we don't know enough today to know exactly how to go after it genetically. It's a very difficult problem.
But I would make, again, the pitch that everybody who's working on autoimmune disease is using CRISPR to understand the fundamentals. I think the advance, I know that this is of less interest to you than the outcomes, but the fact is that I firmly believe, just as the discovery of the structure of DNA, our ability to manipulate DNA in simpler ways, and the use of restriction enzymes have facilitated biomedical research in general. I think everybody who's watching biomedical research with enthusiasm ought to be feeling more positive about the speed with which we begin to understand the fundamental problems that cause autoimmunity than we were before. Question halfway back.
We've been talking mostly about the impact on us as humans, but I've heard a lot about gene drive and doing things where you could wipe out whole populations of species. And how do we control that? Take a mosquito that we don't like, for example, like Zika, and do something that very quickly drives a genome change through the whole population.
Do you want to take that up, David?
Yeah, I didn't mention that. I probably should have. It is a real use for this system. And we could, in principle, get rid of a given mosquito, any interbreeding population that breeds fast enough. You can then basically sending lethal genes through that population. I wouldn't miss mosquitoes if they weren't here.
Well, happily, some of the, for example, Zika only grows in Aedes aegypti. So we could, but I'd like to take your question and make a broader point. it may not have escaped the notice of many in the audience that the CRISPR system comes from bacteria that are defending themselves against bacterial viruses. And yet, as David has described for you, this system works in human cells. I mean, that is goddamn amazing. And this system does also work in other kinds of organisms, in plants, in fungi, and and of course other mammals and potential livestock. So the potential for making some of the changes that we're talking about in human cells in organisms that have agricultural food-producing capacity is very real and is being pursued extremely actively. And I'm very, very glad you brought that up. It was on the list I can't see up here because of the flickering darkness. I think it's something that the audience should be taking away from this, that you make a discovery in one organism, you can use it in lots of others in many cases, and the applications become very broad. David and I were chatting yesterday about the remarkable things that are going on just by sort of capturing organisms, tiny organisms, not viruses, little viruses, big viruses, out in nature and trying to understand the full panoply of what evolution has produced. And we're seeing a lot of very novel things that may, once they're understood, be applicable in efforts to dissect and improve other species. So I think that's a really important factor in all this. I see hands way in the back. I'd like to get that one, but there's one, the guy with the microphone next to him. That's
Hi,
great.
Bert Wells.
Yeah.
Could the two of you address the point and elaborate on the point that Dr. Baltimore raised in connection with what he said he thought should happen in terms of modifying germline cells? And I mean, in particular, what level of understanding is needed to know that it is safe? How much confidence do you have to have in the conclusion that it might be safe to modify germ cells?
Yeah, that's a very good question. And one of the reasons that I emphasized that in dealing with sickle cell disease or Huntington's disease or any of these monogenic Mendelian traits, that we're moving from a disease-causing sequence to a, quote, normal sequence, that is, the sequence that is the average sequence that almost all of us have. And so doing that doesn't really present a challenge to our understanding of the gene, because there's no way in the world that Huntington's disease is a good thing for anyone. There is a situation in which sickle cell disease is good because it prevents growth of malaria in red blood cells, but that's relevant to Africa. It's not relevant to us in the United States. We could get rid of sickle cell disease in the United States and be perfectly comfortable with having done it. Same thing is true with thalassemia and a variety of others. So the single gene diseases, reverting them back to the average sequence, I don't see that that presents us a challenge, a worry about what we're doing. What does present us a challenge is if we start moving alleles around, moving genes around that try to make us more intelligent or more, or taller, or whatever your favorite characteristic would be of your children. Because those genes we don't understand well enough, and there is no, quote, normal for those. It's part of the average, it's part of the variety that exists within the overall human race.
Well, I certainly agree about the distinction between the polygenic traits that we don't understand and the monogenic traits. I take a somewhat different point of view about the safety or the no problem aspect of working with genes like the sickle cell disease or Huntington's. I'd take the stand that we're not ready to do germline therapy for Huntington's at this point. We don't need to use it because we can use the method that David illustrated of selecting embryos and doing pre-implantation genetic diagnosis to avoid the use of gene therapy in the germline. We can do somatic therapy for diseases like sickle, where you don't need to fix every cell. You can fix some hematopoietic some blood marrow stem cells and correct the deficiency. So I never say never, but I certainly agree with the idea that we should be reconvening every few years to think about whether we've reached the point at which we could apply some of the germline technology safely. But right now, I think a moratorium on that is important. One of the things that David didn't mention is that the next meeting that is going to be held by the group that is thinking about this will be in China. We have to recognize that the U.S. has 5% of the world's population. We're a place where scientific advances are likely to occur, but we don't control everything that happens around the world. And I do think that working with Chinese scientists who have done a good job up till now in overseeing the responsible use of these techniques and have been cooperating with the U.S. academies and with the British Royal Society is a very healthy sign. And are there any other hands? There's one hand, the gentleman I said having a chance with the straw hat in the back.
So I wanted to take advantage of the fact that as two Nobel Prize winners, deep understanding of that process, how they decide in Sweden. One question.
Who will get the Nobel Prize?
No, you don't have to mention any names, but when you nominate someone for this discovery, will there be 1, 2, or 3 names on that nomination? And secondly, will the decision in the U.S. Patent Office on who they consider legally the first inventors of this system, will that have an effect on the voting in the Nobel Assembly?
Well, I don't think either of us really want to answer this question. We're happy to declare that time is up, but you yourself were confining the discussion to 3 people because the Nobel Prize is only for 3 people. You might have noticed that the Gardner Foundation awarded prizes for the discovery of CRISPR to 5 people, and I can tell you that many prize committees are spending sleepless nights trying to figure out how to make these awards. One of the things this has brought up, as great discoveries often do, is the fact that not only is there a rich history and lots of people making contributions, but then there are all those postdocs and students doing the great experiments, and it's always hard. It brings up questions of the reward system in science that I think we all find quite difficult to answer in most situations as complex as this one. Thank you very much for coming.
Yes.